And our next speaker, beaming in directly from the land of enlightenment, is Jack Zhang. So Kai did his PhD at the Chinese Academy of Sciences before also moving to the LMB to work with Andrew Carter. And in my mind, most striking as being the sole author on the GCTF paper, which was, I think, cited as Paper of the Year by JSB. When it was released back in 2017. So Kai set up his own lab about 5 years ago in Yale, and he's going to be talking about his work looking at respiratory supercomplexes. So thank you, Jack.
Hi. Good morning, everyone. First, I would really like to thank Renee, Tom, and Laurie, and all other organizers for inviting me to this wonderful meeting. So I apologize for unnecessary troubles traveling outside and returning to the US. And I sincerely hope our world could return to normal in the future, and politicians can make life easier for scientists. So despite numerous challenges, I'm still very excited to take this opportunity to talk about our recent work on mitochondrial respiratory supercomplexes. So I'm pretty sure people may already know about the mitochondrial respiratory chain because it's such a classical topic. The entire respiratory chain, if we include ATP synthesis, consists of 5 main complexes that are embedded in the inner mitochondrial membrane and many clustered along the crystallin. The main purpose of this respiration is to drive oxidation of small reducing molecules like NADH through a series of electron transports, ultimately reducing oxygen to generate water. And the electron transfer process is coupled with proton transport, which creates proton gradient across the mitochondrial inner membrane. This gradient will ultimately power the ATP synthesis to produce ATP. However, for quite a long time, it has been an exceedingly challenging task to obtain these super complex structures and to address questions like whether and in what forms this super complex ever exist. Initially, X-ray crystallography was the main tool to be utilized to study the structures of individual respiratory complexes. Around 10 years ago, structures of the most challenging complex, the mammalian complex 1 was determined by Professor Zazanov's lab and my current collaborator, Professor Jiapeng Zhu, who was a postdoc with Professor Judy Hurst around that time when I was a postdoc with Andrew Carter. In the following years, many individual respiratory complexes were determined at various functional states across different species, as well as some subforms of those super complexes, and even the so-called mega complexes. And for a long time, it has been debatable whether they are functional or how many forms. I mean, these super complexes ever exist within mitochondria. So most structural biology labs would naturally think of using CRI-ET to study in situ structures and ultimately address this question, which was also our original consideration. This technique is like performing a micro CT scan of the cellular or subcellular structures, allowing us to obtain structures directly in situ. Even including amorphous structures, regardless of the resolution, of course. So one major advantage of CRI-ET is that it doesn't require purification of these complexes. Instead, we can directly deposit isolated mitochondria onto CRI-EM grids for direct TEM imaging. So here I'm showing a typical CRI-ET tube series we collected at the very beginning. And the resulting reconstruction. So we could clearly observe remarkably clear like membrane structures associated with all kinds of high density particles, potentially representing various protein complexes. Actually attempted something very similar more than 15 years ago when I was a PhD student at IBP, hoping to obtain some respiratory complexes directly within the mitochondria. But the imaging quality was a major concern around that time. And roughly more than 10 years later, after stepping away from this project, I always thought about picking it up again at Yale when I started my lab with my current collaborator. The conventional CRI-ET approach actually had significant bottlenecks. Here is our original reconstruction using a few tomograms. Of course, I mean, we could, imagine collecting, for example, like 100 times or even 1,000 times more data to improve the resolution eventually. But we reckon there were some major limitations. For example, the samples are already much thicker than typical single particle targets. And the doses we used per exposure were incredibly lower than normal single particle, which ultimately limit the alignment accuracy and high resolution, really high resolution information that could be retrieved from the recorded images. So we thought that some kind of, theoretical bottlenecks. And of course, the environment is so crowded and the membrane signals are so strong that after classification, we frequently ended up with pizza-like structures like this. So to overcome the technical hurdles, we have been developing a method that combines single particle analysis with electron tomography. And our goal finally was to use high dose, high magnification, single exposures allow for high resolution reconstruction. So the goal was to benefit from both single particle and CRI-ET reconstruction and to decompose severely overlapping signals in the crowded environment into a series of simpler non-overlapping images. Then the new images containing one of a few simpler signals that could be processed as if they were obtained from traditional single particle dataset. As part of our long-term goal, we first demonstrated this concept using a relatively simpler system, which was outer arm dining associated with microtubule doublets a few years ago. Of course, it was, complicated enough compared to normal single particle data set. I just mean compared to the mitochondria, it was relatively simpler. So this was used to test the methodology as a proof of concept. The idea was to estimate the microtubule signals and then get rid of the micro signal from the raw micrographs so that we could just treat the data set as normal single particle data set. So this was studied using CRI-ET, before our work. And using this approach, we determined the first structure of dining a complete study, of course, bound to the microtubule and near atomic resolution. We also tested the similar principle in the case of membrane proteins within their native membrane environment with an arbitrary membrane curve model, including those within mitochondrial inner membranes. So here are several examples of the original 2D and also the raw images before and after the membrane signal removal. This is before membrane subtraction. This is after. This is also another example. And it worked pretty good. Finally, in combination of other techniques, of course, there are, a huge of technical bottlenecks we need to deal with using more complicated systems. So we performed extensive data collection and used an iterative particle sorting approach in combination with 2D, 3D, extensive 2D, 3D classification and signal decomposition. And also we benefited from the membrane signals as well. And we first use the strong membrane signals to classify our target, and then we get rid of the membranes, and then we gradually enrich our particles over cycles, and then we just used all the particles we enriched over cycles for the final reconstruction, and then by integrating lots of other tricks, so we finally got really beautiful high-resolution structures of those respiratory supercomplexes. Here is a summary of what we have obtained. The final resolution was pretty good compared to, even better than some of previously reported in vitro isolated complexes. And we could clearly observe like the holes of the aromatic residues. And here is a video my postdoc made to show the densities around complex 3 and also around the Q binding sites in complex 3, and also some sulfur clusters in complex 1. So using the high resolution in situ approach, we could potentially discover as many supercomplex forms as possible all at once, and high resolution. So here is a summary of what we have observed and what we have clearly excluded. The most surprising finding was that we did not observe individual complex 1 and complex 3. We also confirmed that the previously reported mega complex termed type O in our study does exist within mitochondria. That was debatable for a few years. Moreover, we discovered a few other forms such as type B and type X. We also detected even higher orders, but we simply thought they might just represent those complexes, touching each other because they are so close and they just associate with each other. So in the membrane protein field, there are other important questions. For example, how proteins and membranes interact and impact the structures of in their local environment. Previously, people have already demonstrated very powerful approaches to mimic the native membrane environment using, for example, nanodiscs or CRI-ET, or in combination with high-resolution single particle reconstruction and MD simulations. So, for example, this is a very beautiful study performed by my former colleague Alexei, which clearly shows how the distribution of the hydrophobic regions in the supercomplex band, the membrane, is really impressive insight. We use different systems now with higher resolution in situ structures, we could quantitatively analyze the variations of local geometry of the native membrane using directly the experimental data. We observed both common features and the local variations among different super complex forms. For example, there are common features at the heel region of complex 1, and also it shows a clear concave shape around the complex 3 region. We speculate that these common features and variations ultimately impact the diffusion, sorting, and delivery of different ligands and regulates super complex activity. And this was partially, proved using other relatively simpler systems, but not in a complicated system like the respiratory chain. And benefiting from high resolution and native membrane environment, we could easily observe lots of lipids bound to the surface of these respiratory complexes. Our structural analysis indicate that they play a critical role in mediating the lipid-protein interactions. In some cases, such as interfaces between complex 3 and 4, the interactions are entirely mediated by lipids. We didn't observe a direct protein-protein interaction in that case. Because it is in situ, we did not need to use any analogs or inhibitors to lock the protein in a certain state to mimic some reactive intermediates. Instead, we directly observed nearly all endogenous ligands in multiple states, directly with the mitochondria. What is even more exciting is that we found numerous lipids inside the Q binding pocket, which we call lipid curtain. This lipid curtain-like structure is distinct from a normal lipid bilayer that is surrounding a protein complex and the outer surface. It goes inside the protein to form a hydrophobic environment for ligand binding. So the transparent gray is complex 3, and also other adjacent complexes like complex 1 and 4. So it's used to be really challenging to capture Q molecules in previous in vitro studies within complex 3. Especially. So now we know the secret because the lipid curtain was disrupted by deterrents, leading to the loss of native ligands, and because of the loss of the hydrophobic environment, it was very challenging to even mimic the binding of this ligand. Furthermore, we could find that there are specific lipid molecules located in the reactive center and the QI binding site, along with clear protein side chains, native ligands, and water chains mediating the proton transfer coupled with electron transfer. So apparently, lipid molecules are not just providing environment, but they involve in the reaction directly. These were not well addressed previously due to the loss of native membrane environment. But now we could directly observe all this in situ. We also observed the various Q binding states within complex 1 in combination with multi-scale in silicon classification. Conventionally, this goal was achieved basically by using various analogs and inhibitors and extensive CCP-EM classification to sort different binding states. And now we could directly observe all this just using, basically using just one data set in the native environment. We could also capture a series of conformational intermediates of the risky domain of complex 3, coupled with the Q binding states and the QO site of complex 3, and proposed a model to explain how this key domain transfers electrons from QH2 to cytochrome C. It was also highly encouraging that in situ structure studies will almost for sure provide many unbelievable opportunities for pathological, medical, and clinic studies in the future. We show that actually the distribution of the active and deactive complex 1 could be affected by different treatments of the porcine heart. The observation itself was not that intriguing, honestly, as it was quite normal in other biological systems that the protein complex tends to adopt an inactive or inhibited form in responses to environmental stresses, not just limited to respiratory chain. But the real excitement is that we can potentially apply similar approaches to various systems regarding different mitochondrial diseases. So in future, it can be expanded to study potentially a whole world of mitochondrial diseases from different organs with different mutations and different conditions, using different drugs and medicines, treatment during different developmental stages and various animal models, so on and so on. So potentially, I believe structural biology will never see its end because of current structural prediction tools. It's a huge field, and in situ structural biology will definitely open up a new vast field for unlimited potentials, I believe. So I mean, there's lots of fun in the future. So with that, I would like to sincerely thank my collaborator, Professor Jiapeng Zhu. We both did our postdoc research in Cambridge. Jiapeng was Judy's postdoc at MRC and Bill, while I was in Andrew Carter's lab and MRC and B. We started collaborating with each other since I moved to the US. And I also would like to thank my postdoc one, who performed the majority of the actual work. She was Japan's student, joined my lab as a visiting student during the pandemic. I remotely defended her thesis during that time, and then continued as a postdoc in my lab. I would also like to thank my talented student, Chen Xin, who helped a lot with data processing. He just moved to Harvard as a postdoc. I would also like to thank all other lab members and our CCP-EM managers and all my collaborators and my highly supportive Yale colleagues, especially those in structural biology and biophysics, and also our former chairs. Thank you so much. Any questions? It seems I can't hear this.
Hi, can you hear us?
Oh, yeah.
Okay,
Uh,
yeah. These structures are just so amazing. I really, really love it. The one thing I wonder, when you initially show the tomogram of the mitochondria, it looks quite flat, right? So I always wonder, when you
yeah.
plant cells and organelles like that, the surface tension really squeezes on them. So can you see how that affects the organization of the cristae compared to like lamella data?
Yes, indeed. I guess. Yeah, it's quite common that I mean, such big organelles can be easily compressed during the freezing. So, but I don't think it affected the inner memory system a lot. I mean, it it basically affected like I mean, outer memory more than the inner, because mitochondrial inner memory system is so stable, and so I mean, even isolated crystal is stable enough. So I don't think the the local environment was ever affected during the freezing in terms of the inner memory system.
Yeah, makes sense. Thank you.
Yeah.
Hi, can you hear me? Yeah.
Yes.
Fantastic
Yeah.
work. So I have a question about the membrane signal subtraction. So the tools you're using, are they publicly available now? And then
Oh no. So we released the like the microtubule subtraction and the membrane signal removal I mean part, but and we are still developing like the I mean other other part of the entire project. So it's partially I mean released.
Okay, thank you. And also, so I don't know whether this would make sense, but for example, if you do the signal subtraction at the 2D level, and also if you go to 3D and remove the membrane at 3D level and come back to the 2D, what do you think the difference is?
I think the main difference is that we try to recall as much information at the beginning, and using the high magnification, high exposure, single exposure in combination with a tomogram after high dose exposure. So it's already, I mean, exposed and severely damaged, but we didn't care about them, I mean, using the low resolution tomogram. We just use them as like positional and orientation information to guide the decomposition of the single image. I guess that's much difference in my opinion, because theoretically we could record much more high resolution information using high magnification single exposure. That's my feeling.
Thank you.
Yeah.
Does everyone want to go to lunch? One question, Renee.
Hi, Kai, it's Renee here. Thank you for a really lovely talk. I just had a little, and sorry you can't be here because of the climate across borders and things, but had a little detailed question really. Just, did you collect a tomogram after you'd collected your, for every single position? So, you know, you got your 2D image.
Yeah, there was some tricks. Actually, yes, indeed. And the big, of course, this will be ultimately limited by the throughput of the tomogram eventually. So we actually combined, I mean, different strategies. So at the very beginning, I and possibly just a criteria alone, and then hybrid, or we can say strong hybrid by collecting a single exposure followed by a tiered series. And then weak combination, that means we collected single particle and tomograms, but possibly in separate regions, and then just a single particle alone. So gradually you could imagine that we could gradually transfer the information from the earlier state to the later, and ultimately we just improve the resolution using high exposure, single particle data set. In the end, as long as we have sufficient information and references along with the native environment locally.
Makes sense. Thank you.
Yeah.
Great, thank you. So let's thank Jack once again for a very impressive talk. Thank you, Jack.
Yeah, thank you.